Okay, hopefully you can all see that. You can turn your cameras off if you like in the background, so otherwise it comes across my screen. Um, so thank you, Simon, and this is actually my first National Science Week, and I'm really excited to be here actually to talk about my work and the work of my group. Before I begin, I'm joining you also from Gadigal land, and I just want to acknowledge the traditional custodians of the land and acknowledge that sovereignty has never been ceded. I think we've got a lot to learn from Indigenous people, and I want to recognise the strong connection to country that they have. So I'm going to take you on a bit of a journey in the next half an hour or so, a bit of a career journey for one, a little bit of a technology journey, and then also a bit of a journey back in time where we're looking at the key steps leading to the origins of our very existence. So I'm going to address the elephant in the room first, and that is that we're in a pandemic, obviously. And that pun is intended about being in a room because we're all in our living rooms at the moment, locked down. But what I want to do is stress that despite where we are in this pandemic state, a lot of microorganisms are really important and really good actually, and they make up all aspects of life. And I put a little image on the web, which I thought was really cool. All images represent microorganisms, and they are everywhere. They're in all plants, all animals, all. In fact, they're a major part of us. So each of us has around 30 trillion cells, I think, but about 37, 39 trillion microbial cells. So we're only about 43% human. And I hope you'll get this throughout this talk. So microbes are really important, and they literally make the world go round, and they're the reason why we're here. So this is just a little bit. So my science journey, I did a PhD here in Australia, and I was lucky to have a couple of postdocs overseas, over in Germany. I'll just easy for a laser pointer. One over in Germany, one over in Japan. And I'm now leading a research group at UNSW, where we're really starting to answer some pretty profound questions on microbial ecology, evolutionary biology, and this field of astrobiology I'll touch on just towards the end of my talk. So if I was in a room full of library with you right now, I'd ask people if you've ever been fascinated by your ancestors, so tracing back your roots. And it could be anything from really recent if you came here recently, or a few generations back, or obviously a lot further if you have Indigenous heritage. And so this is a shout out to my dad, who I believe is in the audience. This is my tree of life. This is where my dad put this together in a little book he did a couple of years ago. This is my parents here. This is me. This is one of the branches on my tree. This is my son, Ethan. And I have another son. So just a shout out, Dad, this tree needs to be updated. And this is my son here at the Camp Nou in Barcelona. He's actually quite the aspiring young footballer at a tournament he was invited to play in a couple of years ago. And he is making waves, and I am living my life vicariously through him. But okay, so enough about me. So what if I told you that part of what my research group is doing is one of the greatest genealogy searches of them all. That's looking at our great exponentially to the power of what our ancestors, and that is these guys, microorganisms. So this is where it all kicked off, and that's what part of our search is. And before I dive in, I'll try and make sure everyone is more or less on the same page. This is broadly what we call a tree of life, and this has the three domains of life as we know it. So this is the bacteria on the left that I spoke of, all the good and bad bacteria out there. There's another group called the archaea, and these are similar to bacteria to some extent. They're quite small, but they have enough characteristics that they're different enough, and they're really important in terms of evolution, which I'll touch on a bit towards the end. And then we have the eukaryotes, which are us as well. We've got plants, animals, and I said us, we're all up here. And part of my research is also trying to understand how this kicked off, this last universal common ancestor, which was something that was hanging around on the earth, on which all of this tree came about probably 4 billion odd years ago, when Earth was just a wee tiny baby. And now I'm going to literally dive in. I'm really lucky to my area of research, and my field site is in Gathagoodoo country, which is on the western coast of Australia. And these are these stromatolites of Shark Bay. These are these hard rock-like kind of cauliflower structures you can see as we're snorkeling through these systems here. And it's if we ever get out of this pandemic, I hope I can get back there and see these in the flesh. It's like walking back in time. They're absolutely fascinating. Walking through these systems, you feel like you're going back in biological history. And these are living rocks. The name it's a Greek name. Stromatolite derives from the word mattress and rock, and lithos for rock. So living rocks, these geomicrobial systems. Why we're really interested in them, one of the reasons we're really interested, is that their fossil evidence suggests they've been around for about 3.5 billion years. And if you can try and wrap your hand around that time, it's quite a challenge. This is a bit of a schematic. I don't know how easy you can see it there, but where we are right at the end of it, but we got evidence they kicked off around 3.5 billion years ago. And one of their claims to fame is it's thought that they were involved in the oxidation of the early atmosphere. So most of you understand, we know plants, they photosynthesize, which is the process of taking carbon dioxide out of the atmosphere and releasing oxygen. And we all know we need oxygen to breathe, as you all know. But not only plants are important in this process now, but in the past, but bacteria as well, particularly one called cyanobacteria or blue-green algae. And these are thought to be the ones that kicked it off in these stromatolites, probably about 2 billion odd years ago, where the evolution of photosynthesis led to the gradual increase of oxygen in the atmosphere. Eventually it became saturated, and then explosion of complex life as we know it. And then voila, here we are. So you could say we owe our very existence to these little rock formations. And another touch on a little bit towards the end is that we believe that potentially eukaryotic cells, these complex cells, may have even originated in these. So a very quick history lesson on these systems. On early Earth, they're thought to have literally covered the planet, really dominant forms of life. But as higher plants evolved, as I said, they also obviously photosynthesize. So there's competition for sunlight and nutrients with these stromatolite systems. So if you have a plant evolving on early Earth and they're taking over, growing up, taking away and causing shade, that's not going to be great for these stromatolite systems. As well as that, we had evolution of animals, particularly small grazing animals, and these are very nutrient-rich systems. So they would actually graze and eat on these systems. So none of this was great for the stromatolites. So now they exist in what we call extreme environments, where you get high temperature, often UV, salt, really dry or desiccated, and this tends to exclude these high eukaryotes. So the irony is that stromatolites probably kicked off higher life, but higher life actually led to their demise. So they're in restricted environments as we know it now. This is just a really cool image to show what they look like in cross section. You need a diamond sword to cut these in half because they're quite hard. As they're these hard concrete-like structures. I don't know if you can see it where you're sitting, but these are layers. And you can date these like you could the layers on a tree with carbon dating. And these represent deposition. So microbial activity, they're depositing layers over time and they're growing up sequentially. So you can get a lot of information about how old these are, even about the paleo environment that was happening at the time. And this is just some of the cool microfossil images that we see in some of these. So the living stromatolites that we study over in Shark Bay, they have one of the highest metabolic rates of all known ecosystems. So if you take a meter squared of, say, a rainforest, and you measure the productivity, and you can do that by measuring the abilities to sequester carbon, the metabolic rates, and if you compare that to a mat or a stromatolite, the mats or stromatolites win that productivity battle. So they're incredibly important systems, and while I'm all for retaining rainforests, these are also under threat as well. So conservation is really important for these, which I'll touch on in a sec. And so these for us are incredibly important natural laboratories. So we can use these to study processes that potentially may have shaped the biology on the early Earth. And one particular thing is that heaps of activities occur within a stromatolite or within about 1 centimeter. And the activity may have shaped things locally, but even globally. So by changing the atmosphere through trace gases like oxygen or methane. So these systems could have had a profound effect, not just locally, but also globally, particularly on an early Earth. This is just one which I don't have much emphasis on this. Some people ask me, so what else can stromatolites be good for apart from understanding early life and etc. And for me, that's pretty much enough. But there are other potential applications for these systems. But you could use these as biomarkers for a healthy system or an unhealthy system for that matter. Similar to a canary in a coal mine almost, where these systems have been around for so long that if we see problems happening, we can measure changes in populations in these systems. This could be an indication of they're under threat and there's some detrimental environmental impact on these, and that can then have impact on other systems, obviously. And a couple of other ones that we get a lot of novel organisms in these systems, and they can produce novel natural products, enzymes, antibiotics, and we could potentially tap in these. A couple of other ones I just found literally by looking on the web. They've shown to concentrate metal, purify water even potentially, but also oil, their concentration of oil deposits. But as I said, my main focus is more on the basic science that we use these systems for. And so I'm going to give you a bit more insight into the location that I'm interested in, this amazing place on the western coast of Australia. This is just a bit of an aside. I took this picture when I was out there once sampling, and I just thought it was amazing that you actually had to have a sign that's saying that shooting of goats is prohibited. And I don't know if you can see, but there's a lot of bullet holes in the actual sign. So it's a little bit Wolf Creek territory, so I decided to move away from that sign pretty quickly. So the stromatolites in Shark Bay are well recognized as probably the best examples on Earth. There's a few other locations around the world, but these ones are thought to be the most iconic and the most diverse anywhere in the world. The surrounding seawater is about twice as salty as normal seawater, and it's thought that on an early Earth, older and ancient stromatolites also formed in a more high salty water. So again, we're under the idea or the premise that to understand the past, we're using these systems by studying the present. And for those of you old enough to remember, this is again focusing more on the conservation part of these systems. This is the Ghana Climate Change Review back in 2008. And this was commissioned to look at climate change effects on different environments in Australia. And one of them was Shark Bay. And they showed here outstanding examples representing major stages of Earth's history. And these are the stromatolites which I talked about. But they're facing an immediate threat from things such as further sea level rise. So again, really, really important that we're studying these. A couple of years ago, I did an article, if you're interested in the conversation which followed on from this, again, focusing on changes that we're seeing, increased cyclonic events, temperatures, long heat waves, and all of these are impacting not just the stromatolites, but also other systems as well in Shark Bay. Places like the Great Barrier Reef get a lot of media attention for good reason, because it's also under threat with coral bleaching and a number of other impacts. But places like this are also really, really under threat and important for us to bear in mind when we're looking at conservation issues. So again, this is just something that bit up there, and you probably saw the IPCC report came out just last week, showed that many areas of Australia are the most vulnerable, but we're probably one of the countries contributing the least to climate change. So I think we need to certainly act on that. And there's a number of heritage values that I spoke of in Shark Bay. All of these extreme events are increasing more frequently. So we need to understand the effect of extreme stresses on these systems, among others as well. Otherwise, we could get to a point where we get an irreversible tipping point, so where we can't come back to that, and these systems will be gone forever. And this is just an example of some of the things that we can do, and we've done sampling over the last 10 odd years in Shark Bay. Probably 10 odd years ago, there was a big marine heat wave went through, and 5 years ago, big cyclone Olwen, and in fact, there was another one that went through last year, 2020. So we can actually look along on a time gradient to see impacts of these systems, even just over a 10-year period, whether they're susceptible, whether they're resilient. So these are the kinds of things that we can actually look at. And what is a little bit disturbing, literally as we speak, they're planning a titanium mine about 10 kilometers south of this area called Nilamar, where I sample. And this could be a really big problem. We're trying to involved in the monitoring if we can, because if this goes ahead, increase increase in dust from the mine could easily smother these systems or also affect other ecosystems in Shark Bay, massive seagrass systems, dolphins, dugongs, and this could have a real big problem. So we really need to be at the forefront of with the EPA and other stakeholders there to make sure that we're on top of any changes or particularly impacts that we're causing. And I guess my cynicism for the current government is there is no limit, quite honestly. But I hope that there'll be a changing of the guard soon, that perhaps maybe we can make a difference with some things. Another really important aspect of this system I'm really passionate about is Indigenous knowledge and heritage. And this is actually just a saying from the Malgana people in Shark Bay. I won't attempt to pronounce that, but it means this is Shark Bay, you come this way. So it's a welcome there. And so the Malgana Aboriginal Corporation manages native title there in Gathagoodoo country. And the Stromatolites are a really important part of their heritage. And there is one Dreamtime story that talks about Stromatolites as potentially being an ancient serpent that laid eggs, the eggs of the Stromatolites, curled up the body, and eventually water was carved out into rivers and then came back to form springs. And these springs were part of the Dreamtime that provided fresh water for people. So a lot of our work, which is at our science, we really value that, but we're aiming to involve more Indigenous rangers in our field research and try and, as a genuine two-way partnership, incorporate Indigenous knowledge of the different language groups into our research so we can improve our research practices over, particularly at the time that we're involved here. All right, so that was a bit of background into the Stromatolites and hopefully why, you can see why they're important. But despite all that, there's still a lot of unknown questions about how they tick. So my group has been really trying to dive down into understanding at different levels their ecological and evolutionary significance, and a little bit of space science thrown in for good measure, which I'll touch on towards the end. So the other thing which I talk about the different journeys in this talk is my group or my research has also been a walk through technological time. So probably the Stromatolites may not have changed much in the last 10 or 15 years, apart from a little bit through those potential climate change impacts, which I talked about. But our ways of studying them have definitely changed. So our techniques have evolved, particularly into these very high throughput next generation molecular biology techniques that really allow us to go diving down to understand this system at high resolution. This is just a bit of a schematic on how that's evolved, and I'll touch on little bits as I go through. This is actually me sampling there a few years back. So the very early work was trying to isolate microorganisms. So you could take a little bit of mat or stromatolite, grind it up, and you could grow up microorganisms, and you could find out who's there, see what they're doing by growing them up. You could then go doing some very simple molecular analysis, something called polymerase chain reaction or PCR, which involved in a lot of the COVID testing at the moment as well. And this is where you could start to find out a little bit about some functional genes there by doing some very targeted analyses. But the next step is where we're at now, or going even further, which is this next generation sequencing. And this is where you can go down to deep sequencing, single molecules, to work out really what's going on, particularly when you can't get microorganisms into culture. I'll step through a couple of interesting bits that we found out. So this is what they look like if you look at a stromatolite microbe under the microscope. Some of these cool images here, actually interacting with the sediment, as I talked about. They're precipitating minerals. They're making these hard rock-like structures. You get really good visual information with these microscopic techniques, but it's really time-consuming culture. And this is actually just a really cool. Once we grew up one of these bugs, I don't know if you can see it from where you're sitting. It basically attached one of the microorganisms, these cyanobacteria, attached the bottom of the flask. You can imagine like a stromatolite does. And then it grew up in a column of cells, and you can see these little oxygen bubbles at the surface. So this is really cool. It's almost like the precursor of a stromatolite. It was really cool to see. But as I tell you about the technology journey, one of the biggest problems that we have is that you get less than 1% of microbes that are able to be cultured. We just don't know the conditions that all microbes can be cultured in. So many of these unknown microorganisms are there, but we can't get them out. We can't plate them out. We can't look at them under the microscope. So what do we do then? That is when we get to dive down, and that's when all these really cool molecular techniques, we kind of unlock the mysteries of this invisible biosphere, if you like. And this is where we use these high throughput techniques, all automated. This kind of far right, bottom right, if this is coming up for you on the other end, this is meant to represent all the data that's raining down from that we're acquiring now. And the next few slides are just going to go through a little bit of the kind of blood, sweat, and tears from my research group, just to show you the snapshot of the kind of data that you can get back by diving down into this kind of molecular world that we have here. So this is where we can build these massive family trees. Don't worry about the detail of all of this. And this is a big shout out to one of my ex-students now, Alan Wong. This is actually a picture of him over in Romania, doing a postdoc in Europe as we speak, but he's responsible for a lot of the really cool molecular data, which I'm just about to talk about. This is just a tiny, tiny portion of this. I talked about family trees early on. We can look at all the microorganisms there. We can sequence them all. We can now build a tree to see how related they are to each other. We can then also look at what pathways are represented in these organisms to see exactly how they tick. And beyond that, we can actually even reconstruct entire organisms. So there won't be an exam question on this, don't worry, but all of this represents how we can actually take all this molecular data, we can rebuild entire metabolic pathways to work out how these tick. And also this helps us understand about how these systems may have formed in the first place. And it shows us that different organisms are working to the product of one, the end product, of one organism, the substrate for the next organism. So they're working together in a way that is really, really beneficial for the community as a whole. Another one I'll just give a quick example of is survival mechanisms. So I'm just going to drill down a little bit further. So as I said, these systems are in extreme environments. Everything from heavy metals to other extreme conditions they get hit with. You may not be able to see it there, I'm not sure, but this just shows you an example. Looking at the molecular level, there's different survival mechanisms that these systems have, we've shown. One of these is, for example, is the ability to get rid of copper. So copper at high enough concentrations is quite toxic. And we've measured them in Shark Bay, and they're up to about 400 micrograms per litre. Good news is, the stromatolites have got mechanisms to expel copper, so they're coping, they're happy. But other studies have shown that much lower levels of copper can actually have abnormal effects in things such as sea urchins, and even worse from a climate change point of view, that the acidification of the ocean, that's when CO2 is drawing down to the ocean, causes potentially copper toxicity to up even further. So again, understanding survival mechanisms in these systems can help us inform about what might be happening, particularly right next to them. Another quick example, the last biochemical pathway I think I'll throw at you. Nitrogen cycling or any kind of nutrient cycling, really important in these systems, and microbes drive everything. The most important thing to take away from this is that different forms of nutrients, nitrogen for this example, different microorganisms can use different forms in their metabolism. And we found that one particular form of nitrogen metabolism didn't appear to be there, something called nitrification, which is where you metabolize ammonia, this one here. And this is a problem because if you can't get rid of ammonia, if this builds up, this can be quite toxic to any system. And if you have a fish tank, if you have ammonia that builds up in your fish tank, your fish are going to be no more. And so things such as high salt, which we have in Shark Bay, as I mentioned, could be inhibiting the bacteria here. So again, this could be a bottleneck. This could affect other higher organisms in these systems. Again, anything from the seagrasses to other higher organisms in Shark Bay. So again, understanding what's happening in these systems, what's present, what isn't, how they survive is really important to understand holistically all the environment around them. Another way, again, the last one, just focusing on the kind of health, climate change aspect. What we can also do, which is really cool, just stepping away from the molecular bit for 1 second, we can actually measure in situ, which means right into the mat, using these really cool needles, we can measure metabolic activities. And so the ability to produce oxygen is through photosynthesis, as I mentioned. To consume oxygen is respiration, which is what we do and a lot of microorganisms do. And this is just a quick representation of that. And shout out to a colleague of mine, Peter Vischer, over in the US, who comes out with me to do these measurements. So on the left, we have a normal mat, producing oxygen, consuming oxygen. We artificially increase the temperature over in the middle one, and you can see that the metabolic rates are already going down a little bit. And then if you add another layer of complexity, increased temperature and increased salinity, the metabolic rates goes down even further. So it shows that these systems are under stress, and that could be a real problem going forward. So again, it's stressing the importance of monitoring these systems at different levels. And this is just a random aside for you. As scientists, we need to communicate our work, not just like what I'm doing now, but we need to publish our work to disseminate what we found. And in one of the journals I published in a couple of years ago, I managed to sneak in a new word to the lexicon, which is stromatolite. So we have genome, we have proteome, we now have the stromatolite. Okay, one of the last bits which I'll touch on, which is really, really cool. All dark matter you've probably heard of through physics. I'm not going to attempt to explain that because I'm definitely not the expert there. But we have something else called microbial dark matter, which is all these unknowns out there, all these cultured, uncultured representatives. And one of them, which is really cool, something called the Asgard archaea. And the naming of these are found in a hydrothermal vent about 5-6 years ago. It's all, the naming is based on the Norse mythology, if you've ever heard of that. All the gods here: Loki, Thor, Heimdall, Odin, which we think is one of our most recent ones. And this is a big shout out to my student Steph, who just finished her honours thesis last week, and in particular Fraser McLeod here over there on the right, a PhD student of mine, really driving this research in my group, understanding or trying to understand fundamental origins of our existence. So really what we're trying to understand is how this kicked off, particularly the formation of eukaryotes, which is obviously us. And this is just to throw in another quick thread to this whole story, is that maybe the tree of life looks more like this. And this is all these red lines represents the shuffling of genetic material. So microorganisms are great at spitting out their genetic material and shuffling it around by different means. This is a couple of my students here, Julia and Leah, currently doing honours, and they're also diving down different rabbit holes, understanding CRISPR systems. It's cut up DNA, particularly against viruses, different plasmids, circular DNA that again shuffles around genetic material. So this is all helping us understand how these systems tick. So that was a little bit of a snapshot of some of my research at a really kind of broad level. And the question is, what is going on in a stromatolite? And it's like an apartment block, if you could imagine. So you've got different layers, as you would in an apartment block. You've got interactions, you've got exchange of goods and services, waste, communication. The biggest difference is microbes, unlike us, are generally not trying to kill each other. But interactions are really important, working together. Microbes will try and use everything they can to survive, and pretty much every niche you can imagine. And long before Facebook came along, stromatolites and microbes were the original social network. So communicating, literally talking to each other through signal molecules so that they can really make these systems tick and work together. And so these are really, really important systems, understanding, as I said, early life and potentially life elsewhere. And that's the segue into the very last part of my talk, just for the last 5 minutes or so, which is something which I'm pretty sure fascinates a lot of people, and that is whether there is anything at all out there in terms of life. And that's where this field of astrobiology comes in. So this is attempting really, really broadly to address these fundamental questions we've got. How does life begin and evolve? So how does it kick off on Earth in the first place? But then probably the second one is the one we all think about. Does life exist elsewhere in the universe? And the future of life is probably also when we think about our impacts on life on Earth, particularly in terms of climate change. So just briefly, if we think about life on Earth, life on Earth requires water. We all know that. And so one of the things that NASA, particularly other space agencies, do is look for water. Look for water on other planetary bodies, because that could mean that at least the most ideal place to find life, if we're going to find it outside of Earth. And what we do is we look for something called a habitable zone. There's a little picture of Goldilocks. So this is basically the distance from a sun where you can get liquid water on the surface. If you're too close, like Mercury, everything evaporates, water's gone. If you're too far away, everything is frozen. So this is the happy Goldilocks zone where water can exist on the surface. And the other one that could potentially be in that zone is our nearest neighbor of Mars. This is my one joke for the day. Poor one, I know, but is there water on Mars? The answer is yes. The last few years in particular, a lot of this through space imagery, particularly on both poles of Mars, they found there is water, at least frozen water, and almost certainly water beneath the surface. Potentially, they found evidence that water may have flowed on the surface. And so this is the question now, is Mars alive now? And I think it's very unlikely because it's quite inhospitable. I don't think Matt Damon's going to be growing any potatoes at the moment. High UV, very cold. But on Earth, many people have shown microorganisms can survive well below the surface, literally kilometers, but certainly within meters, in the veins of ice as well. So the question is, could there be an accessible subsurface Martian biosphere? And this is where we could look for even evidence of past life. So as I said, particularly with stromatolites, microbes can precipitate minerals and they can leave biosignatures. And that is what's happening as we speak with Percy, the little rover on the planet now, looking to see if we can find potential biosignatures below the surface. And the holy grail, as I've got down here, is sample return, which is on the cards, maybe not for another 10 years or so, but that is hopefully where we're headed once we've had these samples. This is a couple of little cool videos for you. First of all, the launch on the far left. The top right is, I don't know if you can see, this is the little drone that delivers pizzas on Mars. Far left is where, the bottom left is where it's going. This is Jezero crater, and this is just a schematic showing what it may have looked like probably 2 or 3 billion years ago was a delta. And when we see these on Earth, this is a perfect place for microbial life to at least be preserved. So at the moment, this is the last animation here, where Percy is drilling down. And I think it even took some drills last week. That's how up to date we are. And it's going to leave these little cord samples on the surface, potentially for a sample return in about 10 years. That is still to be planned and on the cards, but that is the idea, to get these back and to study these for potential evidence, either of current life beneath the surface, or even past life. If we found evidence of past life in a biosignature below the surface, that would profoundly change our concept or idea of our place, I guess, in the universe. So where does stromatolites, I guess, fit into this whole story? So I mentioned, hopefully from what I've talked about, understanding our own origins with stromatolites. So we can study these systems, both the living ones, which I look at, but also other geologists look at the fossil stromatolites, particularly also in Western Australia, and we can link these together to understand the evolution of early microbial life with its environment. And then in terms of life elsewhere, I kind of mentioned as well, so if we're going to see something microbial life elsewhere, I believe it's more likely to be microbial than green men doing backflips. And as I said, stromatolites or microbial mats, by definition, they're interacting with their environment. They're precipitating minerals, they're leaving biosignatures. So you could imagine on an early Mars that something like an interaction of a microbe with its environment, forming a microbial mat, could leave a signature in the form of these preserved stromatolites that potentially we could find. This could be a long bow, but I think that is, if we're going to find something, could be potentially what we see. And then lastly, away from stromatolites, or a little bit related, is that even if we can't get to a planet, which many we can't get to because of distance, obviously, a lot of work at the moment is looking at interpreting infrared spectra of distant atmospheres, and we can do that by looking at the composition of different atmospheres. So, for example, if you have an atmosphere that has a lot of oxygen or a lot of methane, you can look to see potentially whether that could have been produced by biological means. There's a lot of work you have to do to make sure that something isn't produced abiotically, obviously, but at least it starts to narrow down the kind of candidates you can do by looking remotely for potential atmospheres that could represent something that may have harbored life. In terms of me, I've been really lucky actually to be involved in different areas of astrobiology. I worked in a paper a couple of years ago on microbes that use arsenic, and these may have been really important on an early Earth. Also, a follow-up to a paper on the phosphine in the Venus atmosphere, which I'm a little bit skeptical at what might be happening there, but potentially harboring molecules of life. And even back to our friends, the viruses, we think that viruses might be really important in kicking stromatolites off in the very first place. So I'm ending off in the last few minutes, I know, but I really want to stress a couple of things. One, particularly in this field of astrobiology, but collaboration is unbelievably important. I've had the really fantastic opportunity to work with some really cool and unbelievably intelligent scientists across disciplines, from chemists, geologists, physicists. And by doing that, I'm not an expert in any of those fields by any manner, but by working together, you can actually address these profound questions in a much easier way. And the research, I believe, is a lot better. You get expertise, you get technologies that you wouldn't have accessed just on your own. And this not only improves research, but I think improves the teaching practices that we do it well as well. Not just at the university level, but also at school level as well. So that's why I think collaboration across multiple levels is really important if we're going to advance science in any way. And for me, it's been a really cool experience. It's not just work in a lab. I've been involved in a couple of really cool initiatives, a couple of radio and TV documentaries, which was a little while ago now, was working with a 3D IMAX documentary done by James Cameron, the producer. He went down in one of his little submersibles down to one of these hydrothermal vents, and I was actually the scientific consultant for one of them, where he was asking best ways of sampling that as he was wandering down in the sub. So that was really cool, something I would have never imagined when I started science back in school and then went through university to be able to be involved in something like that, which is really cool. So just to sum up in the last couple of slides, hopefully I've shown that these stromatolites incredibly diverse, novel microbes, but also their pathways. Really important that we manage these effectively, particularly monitor these in the face of climate change, a lot of it by us. But also, as I said, really important working with Indigenous ranges on country to ensure that preservation of heritage is also really maintained. And I guess hopefully that I've shown as a thread is that these systems are really important, understanding our origins, early life, and potentially life elsewhere. So it really is a window into deep time, potentially in multiple directions. And this is just, I don't know, a bit of a couple of profound ones to end off in. This is another big tree of life. I don't know if you can see the details here, but it's not that important. But it's all I want to say is that as humans, we're just a tiny twig on the tree of life, and life is incredibly fragile. So if you're thinking about it, if we were to start all over again, would we get this? And I'm skeptical we would. To get where we started off in the first microbes, all of these events, would have had to occur that to get where we are. It could be the extinction of the dinosaurs, for example. You need a meteorite to hit at that exact same time. So life is unbelievably fragile, and I think that's important as we go forward. That our actions are really important. And I think we need to think about what we're doing to make sure that life is preserved as we know it. Okay, so I'm just going to go quickly, last couple of slides, back to my original story. What is our origin? And I mentioned a couple of my students here. This is Steph and Fraser doing their best blue steel look for the Asgard archaea. And these are the organisms that we believe in our research are involved way back when in engulfing bacterium and eventually forming the first eukaryotic cell, which ultimately was us, and then led to the explosion of life as we know it. So we're looking at this very series of events here. So again, back to our origin, I think it could be an archaea, an Asgard archaea, our very great grandchildren. And I'm paraphrasing Fraser here, and apologies if I get this wrong. He summed this up really well in a talk that he gave a couple of weeks ago. Think about this, that we're all descended from an archaea. All the beautiful things you see, all the memories you've had, is all because an archaea and a bacteria decided that it was better to live together than to live separately. I hope I got that more or less right there. So I did my census last week, which most of you probably would have had to do, and there was an ancestry question. Down the bottom, I wasn't sure if I should do this or not, to put in my other ancestry as Odin, which is one of the archaea. I have no idea if I'm going to get a knock on the door in a few weeks, but anyway, it's good to throw out there where I think we're from. So look, I think hopefully the take-home message you got from this is that microbes rule the world. They're pretty much responsible for us being in here, but also for us still being here. So they clean up a lot of our problems and keep the planet ticking over. So be kind to the environment and to this little pale blue dot of ours, and hopefully we'll be around for a lot longer. And so the research that I've talked about is really driven by curiosity, and generally think it's only limited by your imagination. But the students in my group are what mean everything to me, and they are the reason why I've been able to get where I am. All of what I've talked about are the discoveries that my students have made, and really important to me. So I want to acknowledge those. This is just some of the photos of the lab throughout the year. And I'm really confident of the next generation of scientists that we're in good hands. But for the last couple here, I just want to make a couple of quick acknowledgements. A couple of fantastic students making waves. Alan Wong, again, over in Europe now. Fraser and Steph, a couple of other students along the way here. Fantastic collaborators in the US as well as here in Australia. And all the people that have been involved with my group that have actually made this journey worthwhile. This is a picture of us as my lab group in a biking restaurant actually here in Sydney, just before we entered the lockdown, drinking to our ancestors here. And I hope we get out of this lockdown soon. We can actually do that again. So I hope I've given you a bit of insight into my research world, what makes me tick, what makes microbes tick. And I guess I'll leave where I started, which is the stromatolites here on the left, the shark bay. So this is where home to our great ancestors. I gave a shout out to my dad at the start. This is to my other little boy, Lachlan here. And this is a picture of stromatolite that he drew, which I thought was pretty cool. And so he's the other twig on my little family tree. So I'll probably babble on for too long. So thanks again for everyone for listening. I'm happy to take any questions from the floor. I can stop sharing now if you want, Simon.
Yeah, please. Thank you so much, Brendan. That was amazing. I'm going to pretend I knew everything you were talking about, like everyone else in this chat room. That was fascinating. We actually have some really great questions that I'm going to ask. Melanie asks, and she's written, I'm kind of petrified by the idea of bringing astrobiology samples back to Earth in 10 years' time, especially in light of what we're going through right now with the worldwide transmission of COVID-19. Did you want to talk about that?
It's a good question. So they do a lot in terms of space agency, NASA, whoever. They do a lot of preparation because they're concerned with two things. One is forward contamination, so that's contaminate another planet with us. So every time we send something like a rover or something, there's a potential that could contaminate where it's going. So there's a lot of work done with sterilization so that to reduce the chance of contaminating there. But as you're saying, it's important when you're coming back doing the same thing. So I'm not up with all of the methods, but there's a lot of that is being thought of. So anything that they bring back would be under the highest level of physical containment that they would be looking at. So it's a very good question, but a lot of work is being looked at to make sure that anything that comes back would be studied under the most unbelievable containment that you could do.
Great question from Alan. I'm going to bugger up this word. Stromatolites have survived every climate change due to Earth impacts, mega volcanoes, etc. They have wiped out nearly all of life on Earth several times. So human activities resulting in climate change may well wipe us out, but will it really wipe out stromatolites?
It's a good question. I certainly think that if we keep doing what we're doing and pull the trigger on us, that most microbes will hang along quite happily without us. But again, that's why I stress the conservation part. These systems have shrunk a lot, and if you could see very small changes in sea level, again, the temperature changes that we're seeing and the changes is unprecedented. So yes, they have survived a lot, which is absolutely true. But there is a tipping point that we could be headed towards, that if these get to a point where they can't survive or they're smothered by whatever activities we're doing, then they won't be able to kick off again. Or if they do kick off, it could be in another million years once we're all gone.
Okay, from Liz, she says it was very fascinating. Stromatolite, your word, sounds like it needs a lot of compute power to handle the data. What challenges are you encountering and what's working?
That's a good question because it is really challenging. So a lot of my students are doing just that. So we get all this data, as that image I gave, all this data raining down, and you do need a lot of computing power to be able to dive down and analyze that. And I don't know how best things are working, but a lot of things slow down every time you're trying to do something through different bioinformatic platforms, and the degree of computing power you need means that things slow down. So it can take a long time to analyze all of these genomes and metagenomes we have. So some of the initial steps take quite a while to kind of process that data, but once we have that, then things go a little bit smoothly. So we need to tap into as much computing power as possible. We're trying to hack into some of the top secret, Australian computer network so that we can get all this data as much as we can.
Okay, from Hannah, does the microbial community composition of stromatolites fluctuate over time, or is it relatively fixed?
Another good question. So we've done a little bit, as I mentioned in one of the slides, a little bit of a time series. It doesn't seem. It seems like who is there, as in the different microbes, fluctuate a little bit, but their function more or less stays the same. So in other words, they're pretty resilient, at least over the short period that we've looked at. You might get one group, microbial group, that changes or moves out, but another one will replace it that has a similar function. So their functional stability is probably pretty stable over time.
Okay, great question from Aaron. What are your tips for someone looking to become an astrobiologist? Currently a second year chemistry student trying to figure out my pathway.
Good question. I can do my best to give you some careers. So, as I said, I guess the good thing with astrobiology is that you can work in multiple disciplines. As I said, I'm a microbial biologist or biochemist by trade, actually, but chemistry, geology. So there are people in the if your gig is chemistry, as I think you said, there are people looking at the actual origin of the very first cell. So what chemistry. So there are even at the university that I'm at University of New South Wales. So in this astrobiology field of chemistry is really important to understand. We had to get somewhere from chemistry to biology, and that is where on a very early Earth you had molecules coming together, and that's the part we don't quite understand. So chemists are really important in this whole origin of life question. So there are definitely ways that you could study that in the chemistry field.
And we may make this the last question from Lena. A great question too. Where else in the world are stromatolite colonies exist, and why WA? And is WA has the biggest oldest colony? Does WA?
Okay, so I'll ask the second one first. So the ones in Australia are mainly in Western Australia. In fact, I think they only are mainly on the west coast in Stromatolite in Shark Bay. Some of them are further south near Perth, but that's the main area. Exactly why in that area of Australia is a little bit difficult to answer. One of the reasons why they form that well is because very quickly a sill formed in Shark Bay that caused it to become hypersaline, so they were able to form a bit happier. In terms of elsewhere in the world, there's a couple of cool locations in the Bahamas, Atacama Desert in the Andes in Chile. So there's South Africa. There's a few places dotted around the world, but it's quite limited in their scope. And again, one of the reasons is because we all came along, not just us, but plants, high eukaryotes that caused their distribution to shrink a bit. So the cool thing is that they're all in these great exotic travel locations, which I can't get to because of the pandemic.
All right. Are you able to take more questions? There are a few more coming up. Is that okay?
Yeah, go for it.
Yeah, okay. So why is Shark Bay twice as salty as other areas?
Okay, so that was the bit I just alluded to then. There's a, I can't remember how long ago, 10,000 odd years or so, it was exposed to the normal ocean, but then a sill, which was associated across, it's hard to explain without a map, but came up at the northern edge of Shark Bay, and this caused the inflow of seawater to be reduced, which meant that in the bay water evaporated and became more saline than the outside area. So it was a kind of a physical process that led to salinity building up.
Okay, amazing. Thank you so much, everybody, for joining us, and a big thank you to Associate Professor Brennan Burns. That was amazing, and we're really appreciative of your time. And thank you for coming to National Science Week. I will email everyone tomorrow a survey that the National Science Week hopes that you can fill in for us. And have a great week, and I hope we can see you back in the library by the end of the year. Let's be positive. All right. Have a great night, everybody. Thank you.